Hello, welcome to 13 Universal, and this is A-level CI Biology. We are in exam period. Of course, this is topicals for paper 4A2, Selection and Evolution Series 1. We're starting a series we've done in Gene Tech. This is for selection, okay? So come with me. This is schedule. Remember how we do it. Our basic rules of engagement. This is do it by yourself. Solve this question by yourself, all right? So pause the video as I read the question. You solve it with me. You solve it with me. Guys, let's do this. DNA barcoding is used in species identification, okay? To create a DNA barcode, a specific region, just follow it, okay, of DNA sequence, specific region is sequence so that it can be compared to an online database of reference DNA. One region of DNA that is commonly used is 648 base pairs long within a mitochondrial gene, that's MT gene, okay? Coding for the enzyme cytochrome C oxidase 1. All right, let's see the next thing. The solitary sandpiper Tringa solitaria is a migratory bird. Look at that cute stuff. Okay, let's go on. So DNA barcoding has shown that approximately 2.5 million years ago, T. solitaria evolved into two subspecies. Remember, follow this. One, all right, T. solitaria evolved into two subspecies. Okay, and what are they? Tringa solitaria and Tringa solitaria. So two of them are subspecies of T. solitaria. Okay, they evolved maybe due to mutation. All right, and then we have different. Okay, so a subspecies is a genetically distinct population of a species. So they define it now for you. That has some phenotypic differences, all right, but is not yet reproductively isolated. That means they can still breed to form fertile offspring, all right. They are not reproductively isolated. Come on, let's dive into this. T. solitaria breed in East North America, whereas T. cinnamomea breed in Western North America. So one is in West, one is in East. Okay, East North, Western North. So can you, what does this tell you? All right, remember that we must make that connection between the context, background information, and the content, what you were taught by your teacher, or what your material, your textbooks, the videos you've watched, the content say. Is that okay? So which type of species are usually separated, in different, different locations? Okay, I know you're thinking now. It says suggest and explain how the two subspecies T. solitaria and T. could have evolved from the original T. solitaria population. So this is actually dealing with what? Speciation. So pause the video. What type of speciation, all right, occurs when population, individuals from the same population are separated? They are in different geographical location. What type is that? So that's what they're asking here. They say suggest and explain how the two subspecies. How do I know about that? One is in the northeastern North America. One is in west. So they are separated. Okay. So how do we go about that? Of course, this is talking about allopatric speciation. That's my suggestion. I'm suggesting allopatric. Okay, now I'm to explain it. Why is it allopatric? Because they are separated. They are geographically isolated. So they are separated by a barrier, physical barrier, maybe water or mountains or forests, separated. Okay, and because they are in different environment, so they face different environmental conditions, different random mutations, okay, different selection pressure. And then what happens is that they're separated, and of course there's no interbreeding, there's no gene flow between them, they are not breeding. So this is going to result to different allele frequencies, okay, and therefore differences in morphology, in their morphological biochemical, and of course behavioral similarities. Okay, that's how they became of subspecies. All right, look at it here. Can you see geographical isolation, barrier between population? So there's no low less interbreeding or gene flow. Different mutation occurs, different selection pressure, environmental condition. You can name one. Natural selection. Yeah, that's natural selection. This is natural selection occurring, and this genetic drift. Natural selection and genetic drift cause, both of them cause change in allele frequency. Okay, just that natural selection is due to environment and different environmental conditions, selection pressures, while genetic drift is due to chance. Now, of course, that's allopathic or subspeciation. Okay, remember, if they are not geographically isolated, they're in the same environment. That is sympatric. All right, but now they are geographically isolated. So the 4 marks, guys. Let's run. Fig 3.1 shows an American oyster catcher, Hematopus palliatus. Okay, that is the oyster catcher, cute bird, very fine, long beak. Okay, now says the black oyster catcher, Hematopus bachani, has all black feather. These are all black feathers. DNA barcoding analysis suggests that the American oyster catcher and the black oyster catcher are not, so you have to pay attention, separate species. Suggests how DNA barcoding evidence could indicate that American oyster catcher and black oyster catcher are not separate species. That means suggests, how do we know that they are not separate species? Using the barcoding. Okay, so that's how DNA barcoding. We were told at the beginning that DNA barcoding uses DNA-based sequences. Okay, how does this suggest that both of these species are not separate species? Okay, pause this video, guys. You know the drills. Go on and solve it. Let's see what you got. Okay, let's check your residual knowledge, how you roll this. Okay, how you retrieve information. Pause it. Remember that suggest means give us valid, okay, response. Now, how does this work? Of course, guys, don't forget that how you know that they are not of separate species, that you sequence their DNA bases. Take DNA, all right, material from both of them, sequence it. Their DNA bases shows identical sequence of they're very similar. You want to sequence it, they're very similar. Okay, so maybe the difference in their DNA-based nucleotide, okay, nucleotide-based sequence is very small. All right, so if it is very small, that shows that, you know, of course, they are still related. And if their DNA-based sequence is quite different, all right, I mean, difference is very huge when compared to other species, then you know that, of course, they are just from distance, they are just related. Okay, so see, yeah, see, I mentioned cytochrome C oxidase mitochondrial gene. That's what we're using. Sequence the base sequence of that, and of course, when sequence, if they're identical or very similar, that means they are not separate species. And of course, if there's small number of base different, all right, different, okay, difference or low percentage difference, that means it's separate, or they're different to other species. Difference to other species greater. Okay, 2 marks. Let's go. Custom officers at airports can use a handheld DNA barcode. See, biology is just amazing. Biology is amazing. Solving world's problem, guys. All right, handheld DNA barcoding device. Okay, to identify biological specimens. All right, guys, bringing biological specimens at the borders. Sometimes they bring stuff, organisms that kind of disrupt the food chains, food webs. Some of them are just alien invasive species. Be careful, be warned. But thank God we have these guys called custom officers. So let's go. Yeah. So let's take it again. Custom officers at the airport can use a handheld DNA barcoding device to identify biological specimens entering or leaving a country. Yeah. So just how this handheld, okay, so just how this helps protect endangered species. How does it protect? Okay, remember, you have to pause this video and solve it. Okay, go on now, pause this and try it first. Okay, all right. Welcome back. Now, how does this help? Remember that they can quickly scan, they can quickly, all right, use the barcoding on the organism, okay, and then they will now compare the DNA-based sequence of the organism with online database. When they compare, they find out whether the organism or the product, okay, of an organism is that of endangered species. So if that is endangered species, they can actually seize it, all right, or destroy it, or they stop it from entering the country. They can, sue the person or arrest the person, okay, based on the policy guidance. Look at it here, okay, yeah. So she identifies species quickly, automatically by non-expert. So anybody can do it. Just scan it. Then compare it to red list. Yes, forgot to say the red list is the list of organisms that are threatened. Okay, or IUCN. This red list is IUCN list or site appendix, right? So when you compare it, and then you can either confiscate, destroy, locate endangered species, or prosecute or stop, all right, these activities, or smugglers, traders. So you stop those guys and say, look, what you're holding is endangered species, return it back. Prevent entry of predators. Now this is very important because some of those organisms are predators, right? Because the alien invasive, their competitors, they go on there and spread diseases. So these are the, how it helps to protect endangered species. Okay, prevent predators from entry. All right, so let's go on, guys. um, Look at this. The white bacterian camel, Camelus ferus, lives only in the desert regions of Mongolia and northern China. Okay, I like to go there, guys. I like to know how that place look like. Okay, let's go, guys, let's go. Fig 1.1 shows a white bacterian camel. See that camel? This camel carries load, can survive in desert, is adapted to survive in desert. Can see that. Isn't that cute? Hmm? All right, let's go back to the question. The wild Bactrian camel is at risk of extinction. Guys, is at risk in the wild and is categorized as critically endangered by the International Union for Conservation of Nature. IUCN. There are only 950 wild Bactrian camels. Oh, that's so sorry. Left in their natural habitat. Okay, so just reasons why the wild Bactrian camel has become critically endangered. So this question is asking for threat for biodiversity. If you have the content, if you have done these topics, it's asking you to retrieve information on threats for biodiversity. Okay, threat for biodiversity. So remember, pause this video and answer this question. That's how we roll. Okay, that's how we roll. Now, remember, guys, threats include habitat loss. Maybe the habitat due to deforestation, cutting down trees, affecting it. Not just habitat loss. Climate change can affect it. Global warming. Okay, heat. Okay, presence of diseases. Okay, competition could affect it, or poaching by humans. Look at it. Just memorize it. The questions from these topics are always repeated. All right. Habitat loss, competition with this, or competition or disease from invasive, either farm or invasive alien species. Okay. Climate change, hunting by humans or poaching or killing by humans. Humans kill it and eat it, and they reduce their population. Any of these, okay, threaten them. Any of these could destroy it, kill them. All right. 2 marks. Let's run. Outline the role of IUCN, guys. Okay, what is the role of IUCN? Pause this video right now and try this question. I believe you can do that. Remember that what we do here is that we solve questions, right? That's what we do at 13 Universal Biology. All right, so we solve questions. So IUCN, that's International Union of Conservation of Nature. Okay, so I think what they do is they protect habitat. Okay, all right, and then they advise government on how to protect habitat, and they do a lot of assessment on different organisms to check their conservation status. Okay, so they influence government to make policies right, to protect endangered species by building national parks, okay, right, by making policies against people that destroy um, endangered species that cause them to go into extinction. All right, look at it. See, global, international, worldwide authorities, that's advisory, that's what they do. They give advice to conserve, protect nature. They measure protect nature or biodiversity or habitat. How do they do that? Assess or categorize species, conservation status. They are the ones that produce red list. Red list is threatened. Okay, red list threatened species, the ones that are about to go into extinction, right? Or the ones that are, go into extinction at in the nearest future. Okay, influence government or policy. Yes, educate. Yes, there is awareness to individuals, to the public, right? All right. Some zoos are Some zoos use assisted reproduction techniques such as embryo transfer in their captive breeding programs for endangered species. Embryo transfer has resulted in domesticated dromedary camels giving birth to wild. Yeah, so this domesticated dromedary must be a surrogate mother. There must be a surrogate. Okay, that means they took embryo from the bacterial camel and then put it inside this. Okay, so you see that. All right. Okay, describe the procedure of embryo transfer in a mammal such as camel. So pause the video, describe it. Okay, describe it. Describe it. So now, of course, what you do, of course, is that you can allow mating, right, between two of these bacterial camel, right, and then after some days, you can flush out the embryo, okay, and then transfer it, check, flush out the embryo. That's one of the ways you can source the embryo. You can decide to use IVF to source the embryo. That means you inject hormones into the female and then extract sperm cell from the male, right, camel, bacterial camel, and then allow fertilization to occur in petri dish, then take the embryo, okay? Or you can use artificial insemination also, right, to allow you inject semen into the female of fallopian tube and allow fertilization to occur, and then you flush out the embryo. Then check, these are the ways to source the embryo, to get the embryo to source it. The next thing you do is you check for the presence of check whether the embryo is viable, or check for the whether they have any genetic condition, right? The next thing you do, you now transplant them. If the embryos are viable, they are transplanted into a surrogate mother, okay, that will carry um the, the offspring. Okay, if you don't want to transfer it, you can also freeze it. All right, now see, please memorize it. Also ask question. Mating, these are either you mate IVF, or then remove the embryo from, check, select healthy ones, the ones available. Less embryos. You may freeze it for storage, later use, or implant embryos in different feeding materials. Use surrogacy like they did there. Name female reproductive hormones. You can use, of course, follicle stimulating hormone, right, to stimulate development of follicle, or luteinizing hormone for ovulation, or you can use as well, of course, estrogen for estrogen. Luteinizing hormone allows ovulation to occur. All right, use two of them to stimulate it. Okay. All right. Yes. Then, of course, you can inject estrogen and progesterone to thicken the uterine wall of the surrogate mother. Okay. For the donor mother, use follicle stimulating hormone and luteinizing hormone to start development of the egg cell, the follicle, and the ovulation due to the presence of luteinizing hormone. Then, for the surrogate mother, you treat the surrogate mother with estrogen and progesterone to thicken the uterine line, preparing uterine lining for implantation. Okay, now California salamanders are terrestrial amphibians. An original population of California salamander occupied an area of forest in Northern California. Approximately 10 million years ago, two separate populations, A and B, started to migrate from the original population. So they started migrating, A and B. So from the original population, population A traveled. So they all started from original population. Population A traveled along the coast of California to the west. The Great Central Valley, the west of it. Okay. Population B traveled east of the Great Central Valley. So they moved in different directions. They moved. All right. The two populations now live close to each other in Southern California after migration. So 8.1 outlines the migratory routes of population A and B. See it here, guys. I love this. Okay, this is a migratory route. This is great, okay, valley. So this is migratory route for A. The guy came here and settled. This original population, two of them took off. Say we want to change location. We're looking for new areas to colonize. So population B landed here. Okay, A landed here. Okay, but while they were going, they were separated by this great central valley. So they were not, you know that. They're not interbreeding. Okay. There's no gene flow, and this could have different, selection pressures, and that finally they are closed now. Do you think they will be of the same species or transmission? Okay, let's look at that. Salamanders from population A rarely interbreed. It's going to be a problem. They rarely interbreed, okay, with salamanders of population B. If they do interbreed, the offspring are infertile. So now they have become reproductively isolated. Okay, they used to be of the same species. Now, because of this, they are no longer of the same species because they are reproductively isolated. Suggest and explain the sequence of events that have resulted in these two populations becoming reproductively isolated from each other. Guys, these questions will, you see this particular question, it will keep coming up even this year. Every year they test for this speciation, this type of speciation. Which speciation, involves separation of two populations by a physical, that. So pause this video and try to solve this question. Suggest first and then explain. Sequence, they're asking you sequence of events that have resulted. Okay, now of course you know that there's a geographical, I call it allopatric speciation. So there's a geographical isolation. They're separated by geographical barrier by that great valley. Okay, and then we have different selection pressure, different random mutation occurring. Okay, so we have different, of course, natural selection occurring, genetic drift. So allele frequency changes, so they have different morphological, biochemical features, right, behavioral features, and then that is how they evolve. See that due to great value during migration populations because they are separated, geographically isolated. So no mating because they're separated, breeding or gene flow between them, population, different name selection, pressure, environmental condition, different mutation, natural selection, gene drift, founder's effect. Okay, so so change in allele frequency, all right, change in different gene pool leads to different morphological behavior. But that's allopatric speciation. The one that they are not separated by geographical environment is is called sympatric. All right, let's run. A large number of alpine plant species grow in the mountains of New Zealand's South Islands. Please, I want to pay attention to this. New Zealand's South Islands, alpine plants are defined as plants that live above the tree line. So the alpine plants, they live above the tree line. Now, what is tree line? Which is the height. So tree line is what? The height above which trees cannot grow. So most trees cannot grow here. Why? Why can't they grow here? Okay, most trees cannot grow, but these alpine guys are adapted to grow, right, above the tree line. Now, okay, let's go on. Cannot grow, most trees cannot grow. The current tree line for most South Island is 120 maybe kilometers. Okay, high altitude there. So what are the things that characterize that altitude above the tree line? Okay, maybe there could be strong winds. It's a mountain, thin soil. Okay, maybe, right? There is of course the rocks everywhere. Okay, all right, these are things you should be thinking about. The alpine areas nearest to South Island, they are 1,500 to 2,000 kilometers away across the sea. Many of South Island's alpine species live now nowhere. They live nowhere. This is nowhere else in the world. So they're endemic to this. Endemic means they can only be found here. All right. Explain how a large number of alpine plant species developed on South Island. So how did they develop? Okay. So what is it telling you? How those species develop? How is it that a tree that grows where others cannot grow? How did they develop? Natural select. Oh, guy, shut up, Shandro. Guys, pause this video and let them solve it. I can't help you do this. Come on, you've got to develop this. So how? Explain how did they develop? I've already let the cat out of the bag small, but you start from where? Okay, how? Pause the video and try it. Remember, guys, right? I've already said it. Natural selection. Okay, so that means that most trees cannot survive at that height. So the trees that are adapted, well adapted, survive at that height. Okay, above the tree line. Okay, the selection pressure. Remember when we talk about natural selection, must measure selection pressure. Maybe strong winds, maybe thin soil. So the trees that can survive, all right, now they can survive due to mutation, random mutation, of course, and then we have genetic variation. All right, trees that can survive that selection pressure, their allele frequency increases, they pass on their allele, okay, and then there's a change in allele frequency and change in the morphology, and see, wow, that's a lot. So that's evolution, speciation, natural selection. Any of them, random spontaneous mutation occurs. So some trees, plants adapt alpine high altitude, okay. In this, plants survive were selected. Those that survive selection pressure most likely cold, wow, strong with thin soil. So allele for alpine features passed on to offspring, change in allele frequency to improve population, become morphological observers different, less herbivore competition in new niche. So guys, so you have to connect the context to the content. All right, so let's look at this. Fig 5.1 shows two aspects of history of South Island over the last 3.9 million years. The dashed line shows how the mean height of mountain in Caie region of South Island increased over time. The mountains in this range have a mean height of 200 now 2,400 meters, not 200 at the present. The solid line model is the height of the tree line, okay, the height of the tree line, okay, over time based on geographical climate data. The tree lines, okay, was higher when the climatic was warmer, and then tree line was lower. Okay, the tree line is a line at which they cannot, okay, grow. Was lower when the climate was colder during ice ages. Okay, yeah. Okay, see the dash line. The dash line shows how the mean height of mountains in cloud region of South Alp increase over time. So this is the present time. So that means as time increase, look at it. This is present time. Okay. Yeah. So can you see? Can you see what's happening in time here? So time is from 3.9, right, coming to the present. Okay. You can see that, like they told us, the dashed line mean height of mountains in the cloud region over time, how they change. Okay, you can see that the height from many years ago, the height increased. Okay, so as we move from many years ago to the present, what is happening to the height? The height increased. Okay, then I said, all right, okay, until today. Today is present day. They are 240 meters. Okay, this is where we are today. Where we are. Now the solid, you have to interpret this. Okay, the solid line models the height of the tree lines over time based on geological climate data. The tree lines was higher when the climate was warmer, and tree line was lower when it, okay, can you see it's moving up and down. So if it was higher, okay, higher than what? Okay, can you see solid lines model the height of the tree line over time based on geographical data. So tree line is changing. Tree line is the altitude above which trees cannot grow. So they keep changing. The tree lines are changing. Okay. All right. That's what it shows. Okay. But this is the height of the tree is increasing. But tree lines are changing. They are changing. Okay. Changing, going up and down. Okay. So they're high when it is warm, cold, and low when it is lower when it is colder during the ice age, or this lower ice age. Okay, now let's see the next thing we're told to do. With reference to 5.2, identify regions the with reasons the time period when South Islands alpine plant species developed. Solve this question. If you get it, let me know on on YouTube. We'll send you a pack of chocolate. Try. Reference to this, identify with reasons the time when South Island alpine plant species develop. Okay, positive video. Remember, South pine can develop. They can live at altitude well above 120, okay, 1,200. So let's look at it. Okay, sorry. So if you continue to, okay, the present age, remember from which time, from continue to, right? So don't forget that as time proceeds, the time moves from increases towards the present. So time is decreasing towards the present. Sorry. Okay. These trees are growing. Okay, taller. And of course, we're looking at alpines developed almost recently. Okay, based on what they told us. And of course, they told us that the alpine trees they can grow above 1,200 meters. Now check this. Okay, check this. Right. So, right. So now can you see if you trace this like this? So which ones grow far comfortable, right? From above this. Okay, remember that all of these ones, they are lower. If you trace it, you see it. So starting from somewhere here, okay, all of these ones now they have not reached 2,400. Okay, that is the mean height anyway. But to identify this, we're using 1,200 meters. So if you check now, if you check this one, okay, all of this, this of course, this is tree line increases, one decreases, okay, increases, decreases, okay, it falls up here, right? All right, well here there's a steady increase, okay, all right. But if you check this, you can see that this also, all right, not as high. All right, we're looking at the ones that are far higher, okay, and that could be these from here down, right? From here down, alpine could have developed from here. Somebody asked why not here? Okay, because I'm looking at, all right, the one that has okay, that is quite taller than 100 and 1,200. Okay, all right, and it should be in the most present. Also considering the dash line, that dash line shows how the trees increases. Okay, the mean height of the trees increases. So we check and they find out that it cannot be this is increasing and decreasing. It should be somewhere here. Can you see? Now this is closer. It's closer as compared to all these other ones that are more comfortable. So this, right, touching the dash line and is still also above 1,200. So you can see that if you trace it here, see it goes up and comes down. So it's in between up and down. Up is here 1.0 million years, down is about 0.9. Okay, how do I know 0.9? This is about 10 divisions, gives us 1. So 1 division will be 10 divided by 1 divided by 10, which is 9. So if you count this now, this is 1.0, this is going to, the next one is 9.0.9. So in between them, where this is, okay, is 0.95. So we are choosing this because it's above, far above, okay, all right, enough, far above, so it can survive at this, all right, can you see, can survive. So from here, okay, we started having, all right, to the present time. So this is when we started having alpine. And the alpine developed towards the present time. Okay. All right. So look at it. Time period from 0.95 million to, okay, from that. Okay. Yeah. Why? Mountains high enough to exceed 3 lines. So they're high enough to exceed 3 lines. Remember, they go up and down. Okay. So this is the average of the mountain. Look at the average up and down. This is high enough. Okay. And this provides alpine habitat. Alpine habitat should be above 1,200. Do you get? Yeah. So don't forget that. All right. So if you have questions, you can ask us. Sequence how? Suggest how DNA sequence as DNA. How suggest how DNA sequence data could be used to confirm the time period you have identified. How do you use DNA? Okay, this is used as molecular time to determine the time that alpine trees evolved. Okay, so remember what you do is that you can compare the DNA sequence of alpine and then compare that of maybe the ones that cannot grow up to that stage, that's a lowland species, the ones that cannot grow above 120 meters, right? Compare their DNA sequence. So if the DNA sequence, if the difference is great, DNA, the percentage similarity and difference is great, that means that it's been a very long time. Okay, it's been a very long time. They evolved. They, of course, um, two of them diverged. But if it's a short time, that means they have a more common recent ancestor. But if the difference is great, they have a distant ancestor. Okay, look at it. Compare French similarities. You must use DNA because they say use DNA. Okay, pine trees related and related lowland species or latest species. Find them because they emerge from somewhere. They diverge from a species. Then find time when they diverge. Okay, how do you find the time? By using the many DNA nucleotide, okay, or mutations. You can use the mutations. Means that if you have many of these differences, many nucleotide differences, okay, or many changes or many mutations. If you have many mutations, that means that they have been separated a long time ago, or they have a distant common ancestor. Okay. Now, the rainbow trout Onchohicus macis is a fish that is bred in commercial fish farms. Rainbow trout that have a blue-silver color are sold at higher price than rainbow trout that have a brown. So we have brown and blue. The number of fish with each of the different colors was recorded in a breeding population in one fish farm. Population total is this. Number of blue, okay, is this. Number of brown fish, this is blue, is that 1437, brown this. Okay, so they say the color of rainbow trout is controlled by a single autosomal gene with two alleles, one dominant and one recessive. The blue silver color occurs when a fish is homozygous for the recessive allele, so the blue is recessive. The formula of the Hardy-Weinberg principle states that, you can see that, that this is for allele frequency, this is for genotypic or phenotypic, this formula frequencies, all right? The first one is for allele frequency, second one is use this formula to calculate the expected number of brown fish that are homozygous if the Hardy-Weinberg principle applies to this population. Guys, when you are given to this, pause this video and try it. Okay, pause this video, and try it. Okay, so I want you to see how it rolls. Pause it and try it. 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 All right. Yeah. So don't forget that anytime you want to solve this question, you must always start with the recessive allele, okay? The phenotype, the frequency of recessive phenotype, okay, which is that Q squared there. Can you see the Q squared there? All right. Yeah. Okay. So because the recessive allele, look at it here. Look at it. That's Q squared. This is a phenotype of recessive, the allele frequency for the recessive phenotype, okay? Yes. But you see, for the dominant, there are two of them, okay? Dominant is P squared. This is homozygous, okay? And the heterozygous is 2PQ. It's not easy to solve them because what we can only calculate is dominant and recessive. But to be sure, start with recessive. They always give you recessive, all right? So it's easy to determine the frequency of the recessive. The frequency of the recessive phenotype is Q squared, right? And then of course that Q squared is always the number of the recessive phenotype frequency, which is this is blue. Blue is recessive, right? That's 1437, 1437. Once you find this, you are good to go, okay? Yeah, and then divided by the total number. You know, it's probability, number of required outcome all over the total number. Total number is 2936. 2936. So so you just divide everything. Let's see. So if I say 1004, punch your calculator, try to solve this. 37 divided by 9000, 3000. Yeah, that's 3000. No, that's 2000, sorry. That's 2000 divided by 2936. What does it give us? It gives us 0.48. okay, so Q will give us, you know, square root. So the square will cross to find the square root of 0. Right, 4848 what? 4894. Okay, let me just make it 4. All right, yeah, find the square root. So we're trying to get Q first, so from Q can get. So if I find the square root, or I find the square root of 0.4894, yeah, 94, that gives you 0.699. So Q gives us, all right, that's 0. What? 6996. Okay, so now what will be P? If P plus Q is equal to 1, so P will be what? 1 minus Q. So let's get, and we're looking for Q squared, because Q squared is the homozygous. Remember, what we're looking for is homozygous dominant. Homozygous dominant, right? Homozygous dominant, because brown fish is dominant. And P squared represent homozygous. P squared always represent the frequency of homozygous dominant phenotype. 2PQ represent the frequency of heterozygous dominant phenotype. So P therefore will be 1 minus Q. Remember that Q, we have already found Q, all right, to be 0.6. Remember, 699. Okay, 6. So if you do that, that will give you what? 0.300. Okay, that's 0.3. okay, 300 according to my calculator here, right? Yep. So now you find P squared. So P squared will be what? Because that's the frequency of that phenotype, okay, will be just square this, right? Yep, all squared. So if you square it, what will it give you, guys? About 0.9, right? So if you square it, 0.3 square, that's 0.9. 0.09. So that's the frequency, okay? 0.09, 0.09. But what we're looking for is number. Remember, they said we should calculate the expected number. So we just found the frequency. So you want the expected number, multiply by the population size. The total population size is this. Okay? So that's how you know the number. So multiply the frequency with the population size. 2936. So multiply it by it times 2936, and that gives you what? That gives you 264. 264. They are homozygous dominant. So they will have what is it called? They will have the brown fish. Okay, homozygous, only brown fish. 264. Sorry, this is 64. 64. Right? Okay, let's check. Okay, you see that how we did it? Yeah, so it's the same thing. Let's quickly go to the next question, guys. Okay, some scientists suggested that Hadwan-Bach principle, okay, did not apply in this situation because the rainbow trout were being kept in a commercial fish farm. State two conditions that must be met by commercial fish farm for the Hadwan. Okay, you know the principles that makes it to apply. Pause the video and try it. Okay, what are the principles that make Hadwan-Bach principle to apply? What are the conditions? Okay, of course there must no natural selection, no artificial selection, no migration, right? No of course mutation. There must be random fertilization, and there must be a large population. Look at it. Population is large, no migration, random mating, undergoes sexual reproduction. Organism must be deployed. Yeah, note this. Memorize the always ask question. No selection, no mutation. Okay, 2 marks. Describe how selective breeding can be used to increase the population of rainbow trout with blue silver. So blue silver, how can we? Because blue silver is a recessive. So what we do, pause the video and describe it. So you mate the fish that have the blue silver. When they give rise, of course, take away first, because blue silver is recessive. So take away the brown, and only mate organisms that have blue silver. Do it for many generations because the brown is dominant, right? So artificial selection, human, okay, remove brown, or okay, that dark brown, remove it. Okay, then breed the ones that has blue silver over generation using offspring with this. Frequency of this will increase because only their level will be passed on, guys. So thank you for hanging out with us. All right, so see you in the next episode. I think we'll be doing the next is biodiversity. This is Shedro. I wish you the best of luck as you write this exam. Okay, yeah, remember, practice this video with us. Do not shunt the process. Don't short circuit when you're watching the video. Solve the questions with us. Don't just go see the solution or the answer. Bye. All the best, guys. All the best.